Good morning or good afternoon, as the case may be. Hey guys, and welcome to another lab day. We're going to go ahead and explore some of the evidence for evolution through the marvelous example of the origin of tetrapods. So remember, tetrapods are just four-legged organisms that walk on the land. There are also actually aquatic tetrapods as well, which we can talk about a little bit. But it basically just means a four-legged organism. Tetrapods we usually think of on land are like lizards, the ancestors of snakes that had legs, dogs, cats, anything that walks around on four legs on land. And for a long time, the evolution of tetrapods was really mysterious. We weren't really quite sure where they came from, but there was a general theory that Darwin and many others had proposed that tetrapods that walk on land originally came from organisms in the sea. But which organisms and how did all that happen, right? Because organisms in the sea have fins, and organisms on land have four legs. So that's what we're going to kind of dive into is some of that evidence there today. We're going to take a look first at a little bit of fossil exploration. So take a look at the organisms that have been discovered and some of their traits and features. And compare those, and then we're going to go ahead and actually get into some of the DNA evidence, which is pretty cool. Hopefully by this point you guys have watched the amazing short film, HHMI, on the origin of four-legged organisms, and listened to Neil Shubin talk about his really cool discoveries out there in the Arctic. And so now that you know a little bit more about Tiktaalik, let's take a look at some of Tiktaalik's cousins. So you can read the introduction, of course, but after you get down here to the instructions, we're going to start off by looking at anatomical homology, which again is just homologous structures are bone structures or physical, like musculature and things like that, that we can compare between organisms in order to tell whether they're related. And so we're going to go ahead and do that by using the fossil record, which is our best record of anatomical or homologous structures. So we're going to go ahead and click on this link right here, media for hhmi.org. Again, it is related to the same people that made the video. And we're going to enter into that. That's going to bring us to this really cool interactive that looks at some of the incredible fossils that they have discovered that are related to the origin of the tetrapod. So, it talks about the origin of the tetrapods here. It talks about how fish predate tetrapods because we can find them in fossil rocks that are older than the tetrapod rocks. And so, we're going to look at some of the evidence that fish are, in fact, the precursors to tetrapods. So after you read that, go ahead and click through here. And it's going to bring up the interactive once you click press to begin. All right, so we're going to kind of dive right in here. Okay, so it's going to give us the traits. We can see here these are the characteristics or traits that have evolved in tetrapods and in fish. We can see scales, gills, lungs, humerus, that's that one bone we were talking about that kind of supports the rest of the bones in the limbs. The wrist, you know what that is. Your pelvis, that's the thing that supports your legs. Digits, right? Your fingers, rib cage, you know what that is. Neck and head, you know what those are, hopefully. So these are our traits right over here. You can kind of look over here to look at some of the more key concepts for this, so going to more exploration. If you press on any of the fossils' names, it'll give you the information on those fossils' names, like what the organism was like, what features it had, etc. And we can uncover fossil layers over here. So let's go ahead and dive in. The first thing I'm going to do is I'm going to uncover all the fossils because I want to do that. So da 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 doing paleontological things. Paleontological. How do you say that? Okay, so we have all of our fossils now. Okay, so the first thing I want you to remember is relative dating. Okay, what does that mean? No, it does not mean dating your cousin. All right, come on. It means the relative position of the fossils in the ground can help us to date them by age. So the fossils that are lower down here, such as Eusthenopteron, is older than Acanostega, um, which is older than Pedurps. That's a funny name. Sounds pretty derpy. Okay, so it looks kind of derpy too. Anyway, so the law of superposition tells us that these fossils are older than these fossils, right? And we can even click on them to be able to see whether or not that's true. But the first place I actually want to start is the traits. So here we can see all the traits, and the check marks are obviously the ones that they share. We can see that scales and gills are not shared by two of these organisms, so we can tell that these were probably more terrestrial than fish, whereas these were a weird mix of terrestrial and fish, and then these are fish. Yeah, and then this is also a fish. But this one is actually still alive, which is interesting. Anyway, so we're going to go ahead and take a look at some of these traits. So if you go back here, after you follow the prompts to uncover all the fossils on the left-hand side of the screen, you'll find answers to these questions among the traits, okay? So which bone is shared among all of the tetrapods, and which fishy ancestor has the closest looking bone to it? Okay, and which trait is thin and weak in most fish, but thick and strong in tetrapods? All these questions can be found in the traits. Okay, so rather than give away the answers to these, I'm going to go ahead and go back to this, and I'm just going to open up the traits one by one, and you guys can pause the video and read and find the answers to those. Okay, so hopefully you'll be able to figure out how to do that. This video isn't to give you the answers to the lab, it's just to help facilitate you being able to do the lab if you don't have a device to actually do the lab with. That was probably a very circuitous way of saying, I'm just trying to show you the stuff you need to do to finish the lab. Okay, so let's look at gills. Okay, you can pause the video. Let's look at lungs. You can pause the video. It's cool to me that, that actually the ability to breathe air happened before we actually had terrestrial or land living organisms. That's cool to me. Here's the humerus. I find it pretty humorous. Okay. Here is the digits, your fingers. So there you go. Your rib cage. The neck. And actually the head one is the exact same thing as the neck one. Okay, so there are all the traits that you should be looking at, so use those to answer these first 5 questions. And now that we've explored some of the traits, we're going to look at some of the actual fossils and figure out some things about them. So here are the questions about all of the different organisms, and we're going to use these guys and their names to answer those. So I'll go ahead and do the same thing. I'll click on them, starting with Eusthenopteron, a lobed finfish. Pandurixis, a lobed finfish. With a flat head, too. Look at that. Eyes on the top, helps you to see in shallow water and on land. Tiktaalik, woo! Everybody's favorite. Scroll. Yep. All right. Acanthostega. Bless you. I like this picture. Nah. Okay. Ichthyostega. Dude, that looks like a cool little. I like to have that thing as a pet. It'd be kind of neat. It's like a mix between a dog and a crocodile. Looks like a Pokemon. Okay. Tulirptin. We're getting into the derpy names. Tulirptin. Now, one interesting thing about tulirptin to note is that the only pieces of tulirptin that we found so far, according to this, is the nose, the legs, and the nose and the legs. That's it. We have to guess as to the rest of the body. We could probably conjecture the shape of the head based off of the little bit of the nose that we have, but the rest of it is artist interpretation. But still, pretty cool. Pedurps. I'm sure it's something better than Paderps. It's probably like Paderpes, but I'm gonna say Paderps because that just sounds really funny to me. And also, look at that thing. Tell me that's not Derpy. It actually looks almost exactly like what are those things called in Tatooine that the stormtroopers ride sometimes? What are they called? Not the things that the Tusken Raiders ride. Oh my God, that's gonna really bother me. Okay, I'll look that up later on. That's terrible. I can't believe I forgot the names of those things, but it looks just like that. And the coelacanth. By the way, the Pokemon Relicanth was based off of the coelacanth. The coelacanth was thought to be extinct for a long time. It's a lobed fin fish, but the coelacanth is actually still alive, and we discovered it living deep, deep, deep beneath the ocean. And selected pressures have not really changed it very much from its fossilized forms. We actually have fossils of these things going millions of years back. So these guys have been on Earth for a long, long time. Okay, so use that information to answer these questions. Okay, and this is just some cool bonus content in case you wanted to check out the 3D models of these parts of Tiktaalik that were found in the ground. You can go in and it'll open it up and give you a 3D rendering of the model, which is pretty freaking cool. It's going to take maybe a second or two for it to load on my computer because I'm running so many different applications at once. Start 3D. Whoa. Okay, so here, this is an actual like 3D scan of the fossil of Tiktaalik that Neil Shubin found in the Arctic. And we could see, all right, so here's the orbit, the eye hole, the nares, the nose hole, the brain case, which you feel kind of bad about because, wow, he didn't have a lot going for him, did he? This little bitty brain case. That's pretty cool. You can see the mandibles, you know, the teeth. It's cool to be able to look at this thing's skull and be like, wow, that was a weird looking thing, wasn't it? Looks like a kind of a scary version of the Walmart smiley face. Okay, all right, so moving out of that, that's just some bonus content in case you want to explore that stuff as well. Okay, so we're going to go ahead now. In the video, he said that these homologous traits were really cool and they indicated homology and ancestry. But he also mentioned that DNA says that tetrapods, or things like we're considered bipedal tetrapods, but we have four limbs as well. So tetrapods, he claimed, were descended from fish. And that seems like a pretty strange claim to think that fish eventually gave rise to us, right, and all the stuff on the land. That's a weird claim. Does the DNA evidence support it? He says it does. Will we take his word for it? No, because we're good scientists and we go and research things because we're cool like that. So we're going to go ahead and use something called NCBI, Genbank, and Blast. Ooh, what does that mean? Okay, so this is some kind of complex tools. So I'm going to walk you through it. So what is Genbank and Blast? Okay, well, let's start with Genbank. So you go to Google and you type in Genbank, and it really, you just type in Genbank because, I don't know why they got rid of the second E. Anyway, you go there and it takes you to Genbank Overview, NCBI, NIH, and that takes you to this website. And it'll explain to you what is Genbank and all that sort of stuff. Why should you care and all those things. Basically, Genbank is a library of genes. So I told you guys, and we had talked about and watched those videos about the fact that we have sequenced the genome of the humans. Yeah, we've done that to a lot of things, actually, not just people. We've done that to a crazy number of organisms. And we've stored all the genes that we found here in a giant library. And so we can actually look at them for free, which is cool. So let's go in and we're going to look for a special gene that we can use to sort of help track the relationship between fish and tetrapods and see whether fish are in fact the closest related thing to tetrapods using a gene. Now, what gene do we choose? Because there are like thousands upon thousands upon thousands of genes that we could potentially use. So which one should be used for this? Well, I did some digging. And I came up with a cool little hit on Nature which, if you guys don't know, Nature is one of the premier scientific journal articles. And it says digits and fin rays share common developmental histories. Okay, digits meaning your fingers, and fin rays. And I was like, huh, fin rays are on fish, digits, fish. Okay, so some genes related to digits and fish are probably what I'm looking for. Right, and if you're wondering what a fin ray is, if you look at a fish's fin, these little spines, if you look at, the fish's fin, it has these little spiny things going through it. Those are called rays. And the theory is that those were the precursors for fingers and digits. Okay, so, and we can see over here, this is the coelacanth, again, that lobe-finned fish. It has a humerus. And then, we think the fin rays eventually made this, which is showing that we also have a humerus and that these are kind of our modern fin rays. But what genes actually are shared between digits and fin rays? That was my next question. Well, then I found this awesome article, and this actually has quotes from Neil Shubin in it, that guy who discovered T. pali. And it's from the New York Times, from Fins in a Hand, Scientists Discover a Deep Evolutionary Link. This is a really cool article to show how fin rays may have turned into fingers. So anyway, I was looking through this article and looking for genes, and I found these two. So they talk about when they shut down two genes called HOX13 and HOXD13, the mice that they were testing developed normal long bones in their legs, but their wrist and ankle bones failed to appear, and they didn't grow any fingers. So these were the genes that were related to fingers, and I was like, oh, that's so cool. I wonder if I could find those on Genbank. And so I went to Genbank, and I went to gene because we're looking for a gene here, and I typed in HOXD 13, and I hit enter. And sure enough, here it came, homeobox D13. If you're wondering what homeobox means, when you're developing as a little ball of cells, there are certain genes called homeobox genes, which they basically are, think of them like genetic dominoes. Okay, so you've got all of these genetic dominoes sort of set up to start developing things like feet and hands and limbs and stuff. And as you can imagine, there are a ton of genes that are used to make each one of those. And a homeobox gene is one of the genes that's first activated, and it helps to kind of knock over all the rest of those gene dominoes to make sure that they end up making what they want to make. So when you're ready to make a leg, there's a homeobox gene that's kind of close to where the leg should be developing, like to the little place where it'll pop out. And that homeobox gene will be activated, and it'll cause a little domino effect with all the genes around it, and suddenly, boop, a leg will grow out. So they're very important for the development of limbs and fingers and eyes and ears and all that other sort of fun body part stuff. So anyway, I found HOX13. So I'm going to get out of this tab because I already have HOX13 pulled up over here. So if you're doing this on your computer, just go ahead and do exactly what I just did and get here to HOXD13. You can pause the video if you want, and I'll give you a second or two to do that so you can go ahead and get to where I am. Okay, hopefully now you've gotten to the HOXD13. Let's go ahead and click and dive into this cool thing here. So once we get into the HOXD13, we can see here that its lineage, this is going to be all the organisms that it's in. Wow, okay, so we see eukaryotes, okay, so multicellular organisms, we can recognize that. Vertebrates, okay, so organisms with a backbone, we can recognize that. Mammalia, so mammals. Primates, okay, and then all the way up here to Homo sapiens, humans, right? So all these things in between are just these are lineages that are related to us as well. It says here in summary, this tells us what it does, and these are other names for it. It tells us what it does. This gene belongs to the homeobox family of genes. They encode highly conserved family of transcription factors that play an important role in morphogenesis in multicellular organisms. What the heck does that mean? It's exactly what I said before. They help you to develop certain limbs in your body. And so, it goes on to describe a little bit more about that, but we don't really care about that right now. We kind of know what it does. This will show you where it is on the chromosome and all that fun stuff. We're going to scroll down. Scrolling down, scrolling down, scrolling down. What we want to find is FASTA. Here we go. Okay, so here we're going to look for the mRNA and proteins. If you guys remember, mRNA is going to be what's based off of the DNA, right? In the mRNA, the reason I'm going to click on that is it's going to give us the DNA sequence that the mRNA for this protein is based off of, which is what we want. So I'm going to go in there and click on that, and it'll bring this up. And what's cool is if you scroll down some of this, it'll tell you what certain mutations to this do. So this says it may be responsible for syndactyly. This means different numbers of fingers. So you might have 6 fingers, you might have 4. Let's see, what's another cool one that we can find here? Ooh, interesting. Okay, breast cancer. That's fascinating. Foot malformation. Okay, so feet weren't acting right there. I thought I saw another one earlier here that was pretty cool. But I can't find it anymore. So just scroll down through those mutations. You can kind of see what it's related to. But what we want is the sequence itself. So how do we actually get the sequence? We're going to go to what FASTA. And FASTA refers to just a process of being able to compare the sequence to something else. So here's the sequence. Wow. Now notice it is not an mRNA sequence. It is the DNA the mRNA is based off of. How can you tell? Well, they're T's instead of U's, right? So we can see that that's big, okay? And actually, to tell you the truth, that's kind of small for a gene. Like this is one gene, and this is I don't know how many letters that is in there. It's a lot. But this is just one gene, but it's actually pretty small, which is convenient for us, but kind of makes you think, right? So anyway, we're going to go ahead and copy all this all the way up to the title, and we're going to copy. And then we're going to go ahead and put this into a program called BLAST. Okay, so we have the gene sequence. Now we're going to move on to BLAST. So you can go up here and you can type in BLAST. And if you type in BLAST, it should bring you to this basic local alignment search tool. If you click on that, it'll take you to nucleotide BLAST. Remember, nucleotide is going to refer to the bases in DNA. And since we have just copied a bunch of bases in DNA, that's where we want to go. And we'll go to nucleotide BLAST, and once you're in nucleotide BLAST, you're going to paste that sequence up here. Okay, now, you can kind of ignore most of this stuff. You can ignore all this. You just want to paste that sequence in. But the one thing I do want to come down to right here is this organism box. So we want to compare this sequence to a bunch of different organisms to see which organisms are most closely related to us. Is it fish, like Neil Shubin said, or is it something else? And so we're going to go ahead and do that. And I'm going to actually start off by comparing ray-finned fishes. Okay, so these are the fishes with those fin rays we were talking about, and I want to see how closely related they are to us. So I'm going to click on that. Now, down here, it says highly similar sequences, dissimilar sequences, and somewhat similar sequences. Because fish are not closely related to people, there's a huge evolutionary distance. We're not going to be able to use the highly similar sequences, the megablast, because that's for like really similar sequences. If we did that, we'd probably just get back a bunch of results of like similar human genes or similar genes in primates. So we want to go to somewhat similar sequences, blast in, because if we went to more dissimilar sequences, that would be like the very dissimilar. So we want to go to somewhat similar sequences, you know, close to, but giving us that evolutionary distance, and we're going to hit blast. Now, this is going to take a variable amount of time depending on your computer and the request that you punched in. But it's going to keep refreshing the page, and eventually what it'll do is it will display the results of the blast. Now, in this case, because blast usually does take around usually a minute to 3 minutes to run, I don't really want to sit here and do that for a minute to 3 minutes, so you guys can see. So, oh, well, there we go. Okay, oh, cool. I didn't have to wait that long. That's good. But if it had taken longer, I wouldn't have gone through all that. Well, anyway, now you're looking at this page, and at this point, you're probably like, oh my God, what am I looking at? Okay, all you need to know is that we took the gene that is involved in the development of fingers in humans, and we compared it to ray-finned fish just to see how close they are. That's all you need to know. So we're going to come down here, and now you're like, oh, God, panic. Absolute and total panic. What does this mean? Okay, I'm going to break it down real easy for you, all right? And actually, I'm going to get rid of some stuff over here. I don't care about the max score or the total score or the accession number. Okay, I'll make this easier for you to read. Okay, so over here are the names of organisms that we are comparing the humans to. These are all the organisms that came up. If we're wondering what they are, we can go to one of their names. Let me go ahead and search one of their names here, like Ethiostoma, Ethiostoma spectable. Lo and behold, is the orange throat darter. Okay, so we can see that we can see then that all of these are fish over on the left-hand side, okay, and it's describing what genes it was comparing to. We can see that some of these, they don't say D13, not all of them will. Some will say C13 because maybe that's the closest gene that they have. Okay, so what is this information over here? So we know these are fish. All these are fish. It doesn't matter what kind of fish they are. They're all ray-finned fish. That's what we need to know. Whoop. Okay, so we know these are all ray-finned fish. What does this mean? Okay, query cover means that this is the amount of their gene that lines up with ours. So this one right here, this Hox B13A and our orange throat darter, fish, that only has an 18% coverage, so about 1/5 of that gene is the same as the gene in our bodies. That doesn't seem like a lot, does it? Okay, and this percent identity means to the degree to which this little bit that overlaps with ours actually matches. So about 71 or 72% of this little chunk of their gene that overlaps with ours is the same as ours, or it's 71 or 72% um, uh, identical. So that's that's pretty low, but it's not bad. So let's take a look at this e-value though. Okay, so if we look at the e-value here, the e-value is telling us whether or not we could expect this similarity to be significant. So for instance, does this similarity suggest an evolutionary relationship or does it not? And what's the cutoff number that we're looking for here for that similarity? So the cutoff number is 1e to the negative 10. Anything more negative than that is significant. That means that there's an evolutionary relationship. Anything above that is not significant. So wow, negative 56, that's more negative than negative 10, so that's evolutionary significant. Negative 50, yeah, all of these look like they're evolutionarily significant. In other words, that gene that we're comparing this to, even though it's not exactly like ours, there seems to be some evolutionary significance in the part that is like ours. Okay, so those are actually pretty great numbers. And I could either sort them by query cover, so it would help me to see which ones were overlapped more, or I could sort it by percent identity or by my e-value to see which ones had more of an evolutionary relationship. Okay, so that's pretty cool. So that means that these rayfin fishes, parts of their genes actually are very closely related to the same gene in our bodies, like the fingers. That's pretty cool. Okay, but let's test Shubin's. So things are looking good for Shubin. Okay, he said that rayfin fish are pretty closely related to us, and that looks like it's fairly okay. But let's test it against other organisms. We can't really know how valid it is to say that fish are more closely related to modern day tetrapods unless we compare with other organisms as well. So I'm not going to go through that whole process again because we have to wait. I've actually already done it. So here's my, I've already compared to the rayfin fishes before, and if I press forward, it'll show the results. So this is what we were looking at, before, the exact same thing. I'm going to show you results for other things though. So here are the results. I want to make sure I'm looking at the right thing. Sorry, hang on. Okay, so, all right, there we go. Here are the results for eels. So eels are a more primitive type of fish-like organism, and I wanted to see whether or not the eels were more closely related to modern tetrapods than the fish. And what's the answer? Okay, the first thing we should notice here is that the query cover is much smaller. Okay, so there is a very small part of eel genes that match with our HOXD13, and the e-value is not lesser than -10, it's greater than -10. All of them are greater than -10, which means that there is no significant relationship. So evolutionarily, are eels more related to modern tetrapods than humans? Nope. Let's go on and compare to something else. Okay, so that was the true eels. Okay, and then this one, I think, sorry, we're back to the lobe-fin fishes. I should have done this a little bit better. Oh, okay, so Eptratritus, good God, Eptratritus. See what that is. Hey, these are hagfish. So this is another primitive type of fish. And if we look, okay, so they seem a little bit more related to people than do our ray fin fishes, but not nearly as many. These three, yeah. But for the rest of these, no, their E values are not less than -10. And look at this query coverage. I mean, this is much lower than what we were seeing in the ray fin fishes. So, yep, looks like hagfish are out too. Yep, there's my hagfishes. Okay, and this is, oh, these, I do recognize this. So Latimeria, these are our lobefin fishes, and we can see, wow, yeah, our lobefin fishes are actually pretty good. Yeah, for the most part, all of these from here up, these are all significantly related, and the query coverage gets higher, definitely, than the hagfish. So yeah, our lobefin fishes are more closely related, but our rayfin fishes still are sort of beating them out. And now that might be weird to you because we said that lobefin fishes were the group that actually led into the development of the tetrapod. Remember that here we're looking at the gene for digits, not for the humerus or for the bones that support the arm. So those would probably be closer in our lobefin fishes than they are here. Yeah. And oh, yep, lobefin fishes. And then I think I did an outlier. Yeah, so I'm pretty sure, so Ophiocoma, I think these are brittle stars. Yeah, these are starfish. So believe it or not, in the evolutionary lineage of organisms and in terms of phyla, Echinodermata, which are the starfish, are actually the closest to Chordata, which is we're vertebrates, which are part of Chordata. We are that's our phylum. And so I wanted to see, how closely related starfish were. And if we look, the query coverage is abysmal, and the e-values don't even get close to -10, even though the percent identity of some of these is pretty high. So, but in other words, what we can see is that the brittle stars are not even remotely closely related at all to our tetrapods. So does Neil Shubin's thing about the rayfin fishes, their genes being closer to ours, seem to come out? Yeah, absolutely. Just based off of this very simple search, yeah, we can see pretty clear. Yeah, we are in fact more closely related to rayfin and lobefin fishes than we are to hagfish, to brittle stars, or to eels. So that's pretty cool. And there's lots of other organisms that you could look up. I mean, you could choose anything you wanted to do. And I encourage you to, if you have extra time and you kind of want to play around with this, you can come in here and instead of brittle stars, what if I wanted to compare this to, I don't know, let's compare it to another mammal. Let's compare it to mice and see. So mice, okay. I mean, this should be a lot closer than fish, right? Because mice are mammals and they're also tetrapods like we are. So we should see a much higher query coverage and percent coverage for these. And hopefully it won't take as long because it won't have to search so much. I really hope it won't take as long because, there we go. Okay, and so, wow, oh, wow, okay, now the e-values suck, which is interesting. Oh, here we go. Okay, so here's some good e-values. And the query coverage goes down with the e-values. It's interesting. But we can see that, wow, the query coverage is a lot higher, at least for these guys right here. And it might be because of the type of genes. I also didn't do, I should go back because we should be looking for highly similar sequences And actually, I'm not even going to do mice. Let's go ahead and do, well, I guess we'll just go ahead and say all organisms. Let's see what it gives us for highly similar sequences for that megablast. Uh, this should show us all the primate sequences I was telling you about earlier. This is a really fun tool to play around with. I'd really recommend, and if you're interested in going into genetics, at a later date, and you just want to kind of compare a bunch of genes from one organism to the next, this is a really cool free tool that you can use. And there are better ones that don't cost money either, but you have to download them. So I thought, this might be the best one that we could use. Okay, so now if this starts to take a long time, I might just say look it up for yourself, and we'll go from there, because we're kind of coming up here on a minute, and I don't want to waste any more of your time. I guess I could just say skip ahead to the point at which it looks like on the video I actually got some results back. Um, yeah. Yeah, it looks like it's gonna take a little while. It's taking longer and longer here. Okay, well, for extra credit or just for laughs, you can go in and compare it to just any old organism, or compare it to primates or to mammals or whatever you want to compare it to, and see how closely related it is. And just have fun with it. Anyway, so now that you've gone ahead and seen how I did that, and we can sort of look at it, you're just going to answer the last few questions there at the end, and that'll be it for this lab. So thus far, just to recap, we've seen that homologous structures are in fact shared between organisms. We can see that the homologous structures within the rays of the fins of the fish and the humerus of the lobe-fin fishes do in fact match with the tetrapods, which is pretty cool. And we've seen that their embryology is almost exactly the same, right? Which is, oh, here we go. Here are our results. Let's see what it's, oh, yep, gorilla, and look at that. Now the e-values, again, I don't know why the e-values are so high, but we can see that, wow, the query coverage for the gorilla is 99, the percent identity is 99. The e-value here, I'm not sure, that's really interesting to me, because if we're comparing Homo sapiens to Homo sapiens, the e-value should be really high, because obviously we're evolutionarily related to each other. So I'm not sure what's going on there with that, but we can see that the query coverage is really high and the percent identical is really high. So regardless of the e-value, that should tell you, good God, yeah, we're pretty closely related. Anywho, so we've seen homologous structures are similar between organisms. We've seen that their genes are more similar. We're more closely related to fish than we are to other more primitive types of fish, or even brittle stars and sea stars and echinoderms and stuff like that. So that's pretty cool to know. So yeah, we've looked at some of the evidence for evolution. It's been really cool. I hope you've enjoyed this lab, and sorry for rambling a little bit, but it's a neat lab, and I hope you've enjoyed it. Anyway, I hope you guys have a wonderful couple of days, and wow, I guess I'll see you next week. Crazy to think about. Okay, guys, peace out.